Audio Jungle.
Welcome to today's video tutorial on gram staining. Okay, let's get started. Gram staining. So gram staining is the common, important and most used differential staining technique in microbiology, which was introduced by Danish bacteriologist Hans Christian Gram in 1884. So yes, in the previous video lecture we studied in detail about what is staining. So it is a method of coloring, and so we also studied that why is staining required, and so the different types of staining we studied that there is simple staining where one stain can be used, there is differential staining where many stains will be used. So all that things we studied in detail in the previous video lecture. So here we'll study in detail about a differential staining technique which is called as gram stain. Okay, and it is one of the most commonly used staining method which is followed in most of the microbiological and bacteriological laboratories, and it is the basic. Okay, so I think all bacteriologists will know what is gram staining, and so they would be very familiar in doing it. Okay, so this is one of the basic method, and this was introduced by Danish bacteriologist Hans Christian Gram in 1884. Okay, and then yes, this gram staining is a differential bacterial staining technique. Okay, so as I told in the previous video lecture, differential staining means what? So we mainly do differential staining to differentiate between two group of organisms. So here in gram staining, as the name tells, we do this staining to differentiate between gram positive and gram negative bacteria. Okay, so based on the cell wall composition, we usually classify the bacteria into two types, that is gram positive and gram negative, mainly based on the peptidoglycan composition. So to differentiate this gram positive and gram negative bacteria, we use this gram staining technique. And after doing this staining technique, is both appears in a different color. Gram positive and gram negative appears in a different color so that you can know what is what. And it is the most widely used and most important staining technique in bacteriology, especially in medical bacteriology. It is generally the first test performed on bacteria during their identification and observation process. Yes, this is one of the basic, most important staining technique. And yes, so as soon as you get the bacteria, you want to know about the bacteria, the first method you do is you do staining. You do staining and you observe it under the microscope. Okay, so hence this is one of the basic and most important staining technique that I think all the bacteriologists would know. And the next is the reagents used in gram staining. So yes, as gram staining is a method of differential staining, we use more than one stain here. So first stain is crystal violet. So this appears purple in color, and this is a primary stain as we use it first in the staining procedure. And secondly, we use iodine. Okay, so this iodine acts as a mordant. So mordant in the sense here this acts as a fixing agent. Okay, so that means here it acts as a link between the dye and the target material, that is onto which you want to stain. Okay, and it forms a complex that fixes the dye in place. Okay, so what happens by doing so? The mordant enhances the dye's ability to adhere and intensify the stain. It leads to the more stable and vibrant colors that are less likely to wash away or fade. So that is the function of mordant. And next we do use a decolorizer. Decolorizer means yes, it removes the color here, and that's why we call it as decolorizer. So which will be made up of acetone and alcohol. And then the last stain is safranin. So this acts as a counter stain here. That means, so yes, this is another stain which we use, and the color of the safranin is red in color. Okay, and here staining method is very simple here. So first, yes, we do take a slide. So we take a loop, we make a thin smear of the bacteria. So after that, first we put crystal violet. Okay, so crystal violet is purple in color. Now what happens when we put crystal violet onto the slide? So if there is gram positive cells, if there is gram negative cells, what actually happens is both the cells will take up the purple color. and both the cells will appear purple in color. Okay, and then after this we wash it. After washing, so we'll add mordant. So we'll study the procedure in detail, so just I'm giving a glance of it now. So we'll add mordant, that is iodine. So as I told, iodine helps in fixing the dye to the substance. Okay, so now this actually fixes crystal violet to the substance, and at this stage also both the cells appears purple in color. And then next we add decolorizing agent, so that is alcohol and acetone. So at this particular step, what happens, as the name tells, decolorizing agent, the gram negative cells, no, they get decolorized. That is, the purple color will be removed only in the gram negative cells. So why? Because gram positive cells will have a very thick peptidoglycan layer, and gram negative will have a very thin peptidoglycan layer. So when we put this decolorizer, no, so in the gram positive cells, the stain, that is purple stain, cannot exit the cell very easily. It will not exit. Whereas because gram negative bacteria is made up of very thin peptidoglycan layer, so the purple dye will exit the bacterial cell very easily. Okay, we'll study the principle in detail. So but at this stage, so this gram negative cells, no, they remove the color, they become colorless, and gram positive will appear purple itself. And finally when we do the counter staining, that is we apply the saffron in, we put the saffron in, what happens is, so this remains purple, and this takes up the counter stain, that is the second stain, red, and so gram negative cells appear red and gram positive cells appear purple. Okay. Okay, now we'll study the actual principle of gram staining. And here is a picture which shows you the difference between gram negative and gram positive cell. So as you can see here, this purple color layer is a peptidoglycan layer. So in gram positive bacteria, as you can see, there is a very thick peptidoglycan layer. So whereas in gram negative, so there is a very thin peptidoglycan layer. Apart from that, there is outer membrane also present. Okay, and there is a lipopolysaccharide layer also present. So overall I can tell gram positive will have a very thick peptidoglycan layer, and gram negative will have a thin peptidoglycan layer. Again, this is another picture of gram positive bacterial cell wall, so where it shows the peptidoglycan layer which is so thick. Okay, so this is the difference between gram negative and positive. Now coming to the principle here, see, when the bacteria is stained with the primary stain crystal violet and fixed by the mordant, some of the bacteria are able to retain the primary stain and some are decolorized by alcohol. And the cell walls of gram positive bacteria has a thick layer of protein sugar complex called peptidoglycan, and lipid content is low. Okay, now the thing is, so first, as I told, in the staining procedure we use crystal violet. This is a primary stain for both the gram positive and gram negative. Consider a loop full of bacteria, so where both gram positive and gram negative is present, what happens is, when you add primary stain, that is crystal violet, both the bacteria will take up the crystal violet. Okay, and both will appear purple in the first step. And then after that you will add a mordant, that is iodine. So when you add mordant, what, as I told, so it fixes the dye with the substance, that is it fixes the dye with the bacteria firmly. Okay, so after that, at this step also, yes, both will appear purple in color, because mordant is just making it fix properly, that is it. Okay, and then in the third step what we do, we add a decolorizer, that is acetone alcohol. We add the decolorizer. Now what happens in when we add the decolorizer is, in gram positive bacteria, as you can see here, the peptidoglycan layer is very thick. In gram negative, the peptidoglycan layer is thin. And apart from that, in gram positive, you can also find lipid content is very low. Okay. Now to this gram positive bacteria, when you add the decolorizer, what happens is, see, the decolorizing the cell causes the thick cell wall to dehydrate and shrink. So as soon as you add the decolorizer, this thick cell wall will dehydrate, it will shrink, and it will also close the pores which is present in the cell wall. So small pores will be there. Only when the pores is open, the cell can move inside, that is the stain can move inside, it can come out, right? So what happens here is, as soon as you add the decolorizer, because of the thick layer, no, so it gets dehydrated, it shrinks, and it closes the pores in the cell wall. And as a result, what happens? It prevents the stain from exiting the cell. As a result, the stain, the purple stain which is present over here, no, so it cannot move outside, it will get trapped inside the cell itself, and it still remains purple in color. That is, it does not get decolorized. Okay, that's what. So the ethanol cannot remove the crystal violet iodine complex that is bound to the thick layer of peptidoglycan of gram-positive bacteria and appears purple or blue in color. Yes, so that is what happens here in this term. Third step, when you add the decolorizer, so it dehydrates, it shrinks, it closes the pores. As a result, the purple stain which is present in the gram-positive bacteria cannot move or exit the cell. As a result, at this step also it remains purple. And then okay, so that is now what happens in gram negative cell is that, see, in gram negative also the procedure is the same. You do it together, right? So in the first step, yes, you will add crystal violet. So this gram negative cell also takes up the crystal violet. In the second step, you add mordant. So mordant here also fixes the stain with the bacterial cell. Okay, after fixing, in the third step, what happens is you will add the decolorizer. When you add the decolorizer, what happens is, so here the purple stain, that is crystal violet which was there, will get washed off. So why? Because not like gram positive bacteria, here you have a very thin peptidoglycan layer, and apart from that you have a thick outer layer which is formed of lipids also. That is a difference. So what happens? The stain can easily get washed off. When you put the decolorizer here onto this gram negative bacteria, so it dissolves the lipid layer here. When it dissolves the lipid layer and the peptidoglycan layer is also very thin, what happens? All the stain will easily get washed off. Whereas here it gets trapped, pores only will not be there, and the peptidoglycan layer is also very thick, no, so the stain cannot move away, it cannot be washed, and it shrinks, and the pores also will be completely closed here. But it's not like that here. So this layer will get completely dissolved, and this layer is also very thin. As a result, the purple stain will easily get washed away. And so finally, when you add the counter stain, that is safranin, to both gram negative and gram positive bacteria, what happens? So gram positive cannot take up the counter stain. Why? Because already the purple stain is there. There's no pores, and it cannot take up the counter stain, and it appears purple itself. So whereas in gram negative bacteria, so it can take up the counter stain and it appears red in color. And as a result, very easily you can differentiate the purple color and the red color, and you can easily know which is gram positive and gram negative bacteria. Okay, and here is the detailed procedure of gram staining. How do you do it? So now in the step, see, these are the steps involved. See, first is take a clean grease-free slide and then prepare the smear of the suspension on the clean slide with a loop full of bacteria. Yes, you will take a loop and you will make a thin smear of the bacteria onto the slide. Okay, so after this we will do air drying and heat fixing. So this air drying is so just to dry the bacteria so that you can stain. So you will air dry the bacteria, and then you will heat fix in the sense slightly you will expose the slide onto the burner. Okay, that is not continuously, but so constantly just keeping it onto the burner, taking it away, keeping it onto the burner, taking it away, like that you will heat fix. Okay, why do we heat fix? So that the suspension or the smear which you have made onto the slide has to stick onto the slide correctly, that is firmly, right? And that's the reason you will heat fix, you will air dry. After that, so you will add the crystal violet. See, crystal violet was poured and kept for about 30 seconds to 1 minute and rinse with water. So this is a first step. So you, that is after air drying and heat fixing. Now you will take a dropper and you will add the, or you will pour the crystal violet stain onto the smear. Make sure that it should completely cover the smear. Okay, and then you will leave it for 30 seconds and 1 minute so that the bacteria has to take in the purple stain. Okay, after that you will wash it with water, just running tap water, very slightly you will wash it with water. After that, the second step is flood the grams iodine for 1 minute and wash it with water. The second step is you will take iodine solution, again you will flood it completely onto the smear. You will keep it for 1 minute and then you will rinse it with water. Okay, this is the second step. And then in the third step, then why do we wash here is because to remove the excess stain. Now after adding crystal violet, why did we rinse it with water? In case if there is any extra stain, so by washing the extra stain will get removed. Again, after iodine we are washing here is because to remove the excess iodine which is present. Okay, then after these steps, now in the third step we'll add 95% alcohol or acetone for about 10 to 20 seconds and rinse it with water. So again you will flood it with alcohol or acetone. Okay, and then you will keep it for about 10 to 20 seconds and then you will rinse it with water. And this step is very important here. You shouldn't keep the decolorizer for a very long time to get the accurate results. So you can just keep it for about 10 to 20 seconds or 30 seconds and then you can rinse it with water. And then so after that, so you'll add saffron in for about 1 minute and then wash it with water. So yes, finally the last and final step is you will add saffron in. Again you will keep it for 1 minute and then you will wash it with water. After that again you will dry, you will blot dry, you will just blot to remove the excess stain or water present and then you will observe under microscope. And so by now I think you know very clearly what happens is that when you add this crystal violet, see the cells appear purple in color. Both gram positive, gram negative will take up the purple stain. It appears purple in color. And when you add the mordant, mordant just fixes the purple stain onto the cell, and at this stage also it appears purple. And the third step, what happens when you add decolorizer? So see, these are gram positive cells. Okay, this gram positive cells will remain purple itself. Why? Because they will have a thick peptidoglycan layer, and so they cannot flush off or they cannot wash off the purple stain, and as a result they remain purple. Whereas see these are gram negative, so gram negative will have the thin peptidoglycan layer and the lipid layer will get dissolved and the purple stain will get washed away at this step. Okay, and then finally when you add saffron in, what happens? Only the gram negative takes up the red stain, that is saffron in, and it appears red in color. So whereas this gram positive cannot take up the counter stain and it appears purple in color itself, so that you can differentiate in the same view itself. You can differentiate which is gram positive, which is gram negative easily by differentiating the color, and that's the reason you call this as differential staining. Okay, that is interpretation. That is when you observe it under microscope, what happens? You can easily differentiate which is gram positive and gram negative. That is, gram positive will appear purple in color and gram negative will appear red in color. So here are the two pictures of gram positive and gram negative bacteria. So you can observe, this is a picture of the real specimen or real sample. So and you can see how nicely there is a color differentiation. Gram negative is completely red in color and gram positive is completely purple in color. So this is about gram staining. Okay, so now we'll see some of the important examples of gram positive and gram negative bacteria. So for gram positive bacteria, it is Actinomyces, Bacillus, Clostridium, Corynebacterium, Enterococcus, Gardnerella, Lactobacillus, Listeria, Mycoplasma, Nocardia, Staphylococcus, Streptococcus, Streptomyces. All these are example for gram positive bacteria which appears purple in color. And some of the example for gram negative bacteria which appears red in color is Escherichia coli, that is E. coli, Salmonella, Shigella, and other Enterobacteriaceae, Pseudomonas, Moraxella, Helicobacter, acetic acid bacteria, etc.